The hypothalamus and pituitary form the central command system of the endocrine network. Anatomically, the hypothalamus sits in the diencephalon and forms the walls and floor of the third ventricle. Just inferior to it is the pituitary gland, which is suspended from the hypothalamus by the pituitary stalk and housed within the sella turcica of the sphenoid bone. Importantly, the optic chiasm lies anterior and superior to the pituitary. This anatomical relationship explains why enlarging pituitary masses so often present with bitemporal hemianopia. Functionally and embryologically, the pituitary is divided into two distinct parts. The anterior pituitary, or adenohypophysis, develops from Rathke's pouch, which is an upgrowth of the oral ectoderm. It is a true glandular structure and relies on a specialized portal blood supply to receive hypothalamic hormones. In contrast, the posterior pituitary, or neurohypophysis, is a direct downward extension of the hypothalamus and is composed of neural tissue. It does not use a portal system. Instead, it receives hormones via direct axonal transport from hypothalamic neurons. The hypothalamus controls the pituitary using two distinct communication pathways, one vascular and one neural. The anterior pituitary is regulated via the hypophyseal portal system. This is a low-pressure vascular circuit designed to deliver hypothalamic hormones to pituitary cells in high local concentrations without dilution in the systemic circulation. Here's the pathway. The median eminence is a specialized region at the base of the hypothalamus where neural and vascular systems interface. Unlike most areas of the brain, it lacks a typical blood-brain barrier and contains fenestrated capillaries that allow hypothalamic hormones to pass freely from nerve terminals into the circulation. This structure enables hypothalamic neurons to release regulatory hormones directly into the primary capillary plexus. Blood then flows through the hypophyseal portal veins into a secondary capillary plexus within the anterior pituitary, where these hormones bind to receptors and regulate pituitary hormone secretion. The posterior pituitary, however, operates very differently. Large magnocellular neurons located in the supraoptic and paraventricular nuclei of the hypothalamus synthesize neurohormones. These hormones are transported down axons along the hypothalamo-hypophyseal tract and stored in nerve terminals within the posterior pituitary. When action potentials travel down these neurons, calcium-dependent exocytosis releases the hormones directly into the circulation, allowing for rapid and precisely timed hormone release. The hypothalamus controls the anterior pituitary using a small set of regulatory hormones. These regulatory signals include thyrotropin-releasing hormone, corticotropin-releasing hormone, gonadotropin-releasing hormone, growth hormone-releasing hormone, somatostatin, and dopamine. In response, the anterior pituitary releases a small set of hormones that serve as its outputs. These include thyroid-stimulating hormone, adrenocorticotropic hormone, growth hormone, follicle-stimulating hormone, luteinizing hormone, and prolactin. Some hypothalamic hormones are stimulatory, increasing pituitary hormone synthesis and release. Others are inhibitory, suppressing pituitary activity. Together, they allow precise bidirectional control. What matters most is that the hypothalamus does not simply switch the pituitary on or off. Instead, it communicates information about the body's internal state, such as stress, energy balance, or reproductive readiness, and the pituitary translates that information into endocrine output. A key feature of this system is hierarchy. At the top, the hypothalamus sets the overall strategy. Next, the pituitary translates that strategy into hormonal signals. Downstream, peripheral glands respond and generate feedback. Feedback signals then return to both the pituitary and hypothalamus, allowing continuous fine-tuning of hormone release. This closed-loop system keeps physiology stable despite changing internal and external conditions. Across all hypothalamic pituitary pathways, the same fundamental logic applies. A central signal is generated, that signal is amplified, and feedback ensures stability. A key idea to understand is that the anterior and posterior pituitary operate using two fundamentally different control strategies. The posterior pituitary behaves like an extension of the brain. It is neural tissue, not a true endocrine gland. The hormones associated with it are antidiuretic hormone and oxytocin, and are synthesized in the hypothalamus itself inside large magnocellular neurons. These hormones travel down axons and are stored in the posterior pituitary, ready to be released directly into the bloodstream when those neurons fire. In contrast, the anterior pituitary functions as a classical endocrine gland. It does not receive hormones via neurons, but via the hypophyseal portal circulation. Hypothalamic hormones arrive in the blood, bind to receptors on pituitary cells, and regulate gene transcription and hormone synthesis. This system is slower, but it allows for signal amplification and integration. Very small amounts of hypothalamic hormone can generate large changes in pituitary output, which then drive powerful downstream endocrine responses. So, the pituitary is not a single functional unit. It is two control systems sitting side by side, one neural and fast, the other endocrine and regulatory. The pituitary does not simply respond to hormones being present or absent. Instead, it interprets patterns of signaling. One of the most important principles is that the hypothalamus can control pituitary output not just by stimulation but also by inhibition. The clearest example of this is prolactin, which is under constant inhibitory control from the hypothalamus. When that inhibition is reduced or removed, prolactin levels rise, even without any active stimulatory signal. Another critical concept is pulsatility. Some hypothalamic hormones are released in pulses rather than continuously. The pituitary is sensitive to the frequency and timing of these pulses, not just the absolute hormone concentration. Continuous exposure can actually suppress pituitary responsiveness, while physiological pulsatile release maintains normal function. The pituitary also acts as an integrator. It receives multiple hypothalamic inputs, peripheral feedback signals, and internal modulators, and combines them to adjust hormone output appropriately. This is why pituitary hormone levels often reflect the overall physiological state of the body rather than a single isolated signal. In this way, the pituitary functions less like a simple relay and more like a decision-making hub, translating hypothalamic intent into coordinated endocrine output. Rather than memorizing individual endocrine diseases, it is far more useful to understand a simple framework that applies to all hypothalamic pituitary systems. Every axis follows the same logic. A signal is generated, a response occurs, and feedback adjusts that response. When interpreting hormone results, the first step is to pair each pituitary hormone with its target gland hormone. Once you do that, three broad patterns emerge. The first pattern is intact pituitary drive with a failing target gland. In this case, the pituitary hormone is elevated, but the peripheral hormone remains low. This tells you that the control system is functioning, but the gland itself is not responding. The second pattern is loss of central drive. Here, both the pituitary hormone and the peripheral hormone are low or inappropriately normal. This indicates a problem at the level of the pituitary or hypothalamus, where the signal is not being generated or transmitted correctly. The third pattern is loss of feedback control, where hormone levels remain high despite signals that should suppress them. This suggests autonomous hormone production somewhere along the axis. Importantly, disorders of the hypothalamus or pituitary often affect multiple axes simultaneously because the problem lies in the control center rather than a single target gland. If you approach endocrine problems using this signal response feedback framework, the system becomes predictable, logical, and far less intimidating.